The overall goal of the following experiment is to observe the cytotoxic effect of the phenanthridine derivative PJ34 during mitosis in multicentrosomal human cancer cells. This is achieved by labeling centrosomes with GFP gamma tubulin and chromosomes with H2B red by transfection. The goal of transfection is to enable tracing migration and clustering of centrosomes and arrangement of chromosomes during mitosis in live multicentrosomal cancer cells. As a second step, some of the transfected cells are exposed to PJ34, a molecule that acts as an extra-centrosome declustering agent. Next, transfected live cancer cells are scanned by confocal microscopy in order to capture events during mitosis associated with centrosome and chromosome migration, rearrangement, or clustering. Sequential images that are obtained show the declustering and cytotoxic effects of treatment with PJ34 in cancer cells during mitosis based on confocal imaging of live cells.
The implications of this technique extend towards a targeted cancer therapy exclusively eradicating extracentrosomal cancer cells during mitosis. Demonstrating of this technique will be our collaborator, Dr. Castiel from the Cancer Research Center in Sheba Medical Center, and Dr. Leonid Mittelman from the